Okay, we are starting. It's 2 o'clock, so let us begin. And I just show you what we're going to be sort of ping ponging a little bit between the genetics chapters, which are conveniently chapter 8 in both books, our Cowan textbook, and which looks like that, and then the one that says chapter 8 lecture. This is Nestor's textbook, and it is this one. So because they have different illustrations and different ways of approaching the same materials. That's what we're going to do. But first, depending on how conversational you are with the folks who were here before you, I anticipate a lot that this is what I am doing after this rather unpleasant test experience. So I have picked out 28, count them, 28 of the world's worst answers to these are questions that approximately half the class missed. Okay. It is stressful. These are terrible. I mean, to me, these are questions that half the class missed. So they're awful questions, not the questions, the answers. That's a better way to express it. So they're the same questions you just had on the test. Okay? But I've got 28 of them. If you can complete them all successfully, for every 2 you get a point on top of, and then I'll adjust the grades appropriately. Because you all need it. Not all, but collectively you all need it. Because and look, we need to do better on this. So at some juncture, when everybody, and Russ hasn't taken it yet, so he's not going to look at it. But But I can go over some of because you need some help in test taking technique and reading questions appropriately, because you don't. That's I can't deal with the stress part. Okay, that's test taking anxiety is people have, but I can't. That's not my area of expertise. To me, the thing that deals with anxiety effectively is preparation and confidence. To me, and that comes over time as you learn what to do. Because when you start to take successively more difficult courses, it requires a certain amount of preparation and a certain amount of approach once you get to know what the style of the teacher's questions. So that's why I'm affording everybody this opportunity. Again, some of the students who had had me previously already had a heads up, but that's the way it is. Some of you would have, but I didn't happen to teach any AP1 this year. Okay, it's just with who gets assigned what in the department, and different professors are doing different things. It's just the way it works departmentally in any school. Okay, so you choose things, and to some extent it's based on seniority. And while I have that in age, I don't have that with academic rank. I'm older, but they are higher in rank than I am. It's okay. I've gotten over it. I'm fairly clever. Not that they are not. So on your way out, not now, grab a Scantron and grab this. Let's get them back on Wednesday, if at all humanly possible. Okay, and we will be able. Look, if somebody did great and got an A, you don't have to do it. I mean, you can't get a higher A. It doesn't work that way. No. We'll wait. We can turn. You'll see it. Nice to see you today.
Hi.
Take Friday off?
Yeah.
I know. Early weekend.
Yeah.
See, my powers of observation are not acute, but not necessarily obtuse. I think it's the right word. All right, everybody. So that was the last class took 5 minutes to explain this. Now it's 4. Now that you're in a better mood with me, okay, picture of the day. After my son, that one, not this hat, but this without the hat, has been at sea for 14 of the last 18 months. Just got home after 4 more months of deployment. So, and son, grandson, granddaughter, wife. Here, that's a destroyer in the background. So they got in the pouring rain. Yeah, in Mayport. It was wet. They canceled Daytona. Remember, for you NASCAR fans, they canceled Daytona. I mean, Daytona is what, 50 miles away from Jacksonville, maybe something like that. So that was the picture of the day. So there's another one here. See, my granddaughter. Yeah, thank you for the appreciate that. Those are good. It keeps me in a good mood in the class. Okay. So let's begin. This is I didn't start genetics with you, but I will, in an effort to try to give you some ideas. The biggest problem we have, I think, is particularly these emergent forms and mutations, and really that's what we deal with. There's three vis-a-vis this test. There are 3 chapters on the next test. The one we just completed on, germicides, antiseptics, disinfectants, radiation, sterilization. There's the virus chapter, which I think is interesting and very clinically oriented. So we start to learn about the diseases and how they're characterized. Not so much on what we do about them, but you started getting our hands into them, how they're spread particularly, how we categorize them, as I said before. This is the one I do the least of on the test. If there's 65 questions, there are only 10 or 15 from this chapter. So, I mean, I've tried to make them as simplistic as possible. My major concerns are to understand why prokaryotes, specifically bacteria, viruses to a lesser extent, and it's a different reason, because viruses, you're going to find when we do viruses, RNA viruses, are inherently unstable genetically. But why bacteria are so much more likely to mutate than our cells. And I'll start with the 3 big reasons, and then I'll carry on with the rest of it. So I figure it's always good to have a starting point, and we might as well start here. And so I'll go over that learning outcomes stuff. Sure. So think of it this way. Our cells, on the average, are less likely to mutate compared to a bacteria by an order of somewhere between 1,000, maybe I didn't say 10,000, maybe 1 in a million or even 10 million times. It is an immense amount. Our cells have a lot of genetic safeguards. And I'm not just our cells, but eukaryotic cells in general. So there's 3 reasons. Eukarya versus prokarya, which is bacteria. Eukaryotic cells have pairs of chromosomes. Prokarya have a single chromosome. The DNA is the same. It's double-stranded DNA. Called DS double A, DNA for double-stranded. It's made of the same A's, T's, G's, and C's. Nothing unusual about that. Why the difference? What the difference is this. This single circle, when you're working with a lab on E. coli or Staph aureus, typically what they give you is a fairly generic form. That is, and because it displays the normal characteristics, we use the term wild type. If you get one that mutates, or you find one that's in a disease situation that has a mutation. And what's a mutation? Mutation, very simple definition. Error in DNA. That's mutation. What the mutation does, does it cause cancer? Does it cause somebody to die? That's a whole different thing. It's a birth defect. So not all mutations are created equal, as you can see. So when you say mutation, the definition is very specific. So a normal one without a mutation is called the wild type. Everything else, anything that's not the wild type, is called a mutant. So if E. coli is irregular, effectively it's a mutant strain. Okay, we see that relatively frequently. It doesn't have all its characteristics. But you and I, we don't see that. Why? Well, we'll start with this, pairs of chromosomes. So we all have With 23andMe, Ancestry.com, all those 23 pairs of chromosomes. What does that mean? Well, you have 4, either one from mom, one from dad, on either side, and you only have a certain limitation. We don't even use the term wild type of mutation. Very famously, you all know this, we use dominant and recessive, or recessive and dominant. We're both dominant or both recessive. Oh. And what's the idea? What makes dominant dominant? If the dominant is present and the recessive is present, the dominant is expressed. So if we have a mutation on the recessive strand, who cares? It's not going to be the one that's going to make all the proteins the way we can identify it. So immediately, pairs of chromosomes. What if we have 2 dominants or 2 recessives? Well, again, we only express one portion of the chromosome or the other. We don't express both. So if it happens to fall, it's a 50-50 chance that we're going to express it. So immediately, the mutations are less likely to be problematic. Okay? Just because of that. Bacteria does not have a pair. It's got a circle. So wherever the mutation is, it's a problem. Now, not done yet. Something that you probably didn't do in AAP, you might have, and I don't typically spend a lot of time on it. Basically, it's what we call transcription. And this is why we have to go over here. We have DNA. DNA undergoes transcription. Makes messenger RNA. Messenger RNA eventually makes proteins. That whole process famously is referred to as translation. See, transcription is basically an exact copy. If I dictate a chart, a medical transcriptionist in a hospital will listen to my recording and will type exactly what I say. Translation means what it actually means. Let's say if it's in a different language in English and someone wants to say it in Spanish. Obviously, they would translate. It wouldn't necessarily be identical words. It would just give you identical meaning. So, in the process of doing this in bacteria, this is how it works. DNA, so if there's an error here, there's an error here, and potentially there's an error. There's no way for that error not to be there. There's a mutation in DNA, it's going to be an alteration of messenger RNA. Depending on where it's located, it might be something that affects the protein. In our cells, there's a different step. It's called messenger RNA processing. In our cells, that strand consists of alternating segments. Okay, and those segments, I'll draw big lines, I don't have another color to do, but I'm not using it. Handy. And those segments are called introns and exons. You don't have to know it so much, but just understand what they do. So the big I, introns, is short for insertion sequences. So only in our cells, and there's a lot of introns. In eukaryotic cells, only about 2 or 3% of that DNA actually makes the proteins that give us our skin color, our hair color, our eyes, our freckles, whatever. Limited amount. The rest of the DNA, the other 97, 98%, is what we call regulatory. It basically makes the system work properly. So we're all the same other than maybe a couple of percentage of duration. So, I mean, it just kills it. Oh, you're special, you're not. To me, genetics means it gives us some advantages and some problems. Like in the world, 100% of people are more athletic. As much as I dislike that. So here you have these I's, insertion sequences. What we do is we get rid of them. So we have a little X here, an E here called an exon, and another one here, and we splice them together. And so effectively what we have done is we have taken that original strand and shortened it substantially. If the error was on that strand that we removed, the intron, no problem. So that's another safeguard as well as pairs. And the last safeguard is an enzyme that they have in the PowerPoint. Eukaryotic cells, let's just put you, have something called DNA polymerase Roman numeral 3. Bacteria have DNA polymerase 1. So again, what's a mutation? Mutation's an error in DNA. And so DNA is pretty easy. Remember, it's A's and T's, G's and C's. Everybody remembers that. Adenines are with thymines, guanines are with cytosines. So if we have an adenine and a thymine, guanine and a cytosine, it's a good thing. If we have a guanine and a guanine, something is wrong. That's a mutation. Okay, I know you're on the wrong side. So DNA polymerase 1 in bacteria comes along and goes, oh, A, T, G and C, G and G. One of these is wrong. Okay, one of these things is not like the other. No Sesame Street. And it's as likely to move the right one as the wrong. 50-50 chance, mismatch. Our cells, DNA polymerase 3, T, G and C, G and G. What it does is not only recognizes the defect, but removes the mutation replacement. It's a much more sophisticated mechanism. Those are the three things. Here's your chromosomes, messenger RNA processing, DNA polymerase 3 that make eukaryotic cells less likely to mutate. Moreover, you and I go into a room and 63 people were in there smoking and they didn't have the windows open. Okay, you're being exposed to nasty things, carcinogens. That'll cause mutations. For most part, even if it created a mutation, we make 1 billion cells every day easy, maybe more. There's always going to be a mutation. These mechanisms capture that. It's all about numbers. You have so many repair mechanisms, they catch these problems. Bacteria don't have that for those reasons. So if they are exposed to a mutagen, if they are exposed to a toxin or an adverse condition, something that's going to cause a mutation, the likelihood is we are going to see an increase in the mutation rate by a factor of 1,000. So the difference in mutations are spontaneous. So those billions of cells we make every day, there's going to be a few errors. If you would do 1 billion things and get 999,999 correct, whatever that is, whatever the number is, 99,999,999,000, you make one mistake. Not a big deal. Everybody be happy. Okay, and moreover, you can correct it. But if you turn that number, if you make 10,000 bacteria and 10 of them are mutated, now we have a problem. Because sooner or later, those are going to survive and create problems. So you're going to see the difference between what are called induced mutations when they've been exposed to a mutagen, X-rays, toxins, chemicals like we learned about in the last chapter, being ones that are spontaneous, basically the price of doing business. Because it's a living system and living systems make mistakes all the time. That's sort of the jumping off point for what we're looking at here. So they're going to look at terms you hopefully do know. Your genome is the assembly of all your genes. Your genes are, and we have about 30 to 50,000 in our chromosomes that are markers that govern certain characteristics or processes, more or less. It's not just like one isolated area, it's usually a series of things. And so when we say genotype versus phenotype, genotype are the actual genetic markers. Okay, so maybe the best one to see of that, even though it's not dominant recessive per se, is blood typing. I mean, it's easy to test. Are you A positive? Are you B negative? It's a very specific, that's your genotype. Okay, so it's something like that. But when we talk about phenotype, we all might have similar genotypes. Our phenotypes are something that we can observe and measure. It's more of a so that blood was probably more phenotypic than genotypic, if you want to think of it this way. So what does that mean? Eye color, hair color, you know, surprisingly long, skinny fingers are not genetically as common as short, stubby fingers. Okay, those, remember they learned about these things, dangling earlobes? Is it ringing a bell? That type of stuff. Yeah, our earlobes dangle more than ones that are attached. You know, a small percentage of us have that, you know, that hitchhiker's thumb, that kind of a thing. Those are common little things you look for in genetics classes, basic ones. And they're there. But how do we measure them phenotypically? So you're going to see that in the lab with E. coli. You're going to do a lab where you plate it out on something called EMB agar, which is an agar that's designed to grow E. coli and similar organisms. And how are you going to tell if E. coli is there? It's going to change colors. It's a very, so it's identified in a visual or growth kind of a way. You're going to be able to see it in some metabolic fashion or some visual fashion. So that's a phenotype. And basically we're talking about something called a lactose fermenter. So all that. So that's what's going on. And the basis, obviously, of heredity. So if you're fortunate enough, you most people will see similarities hereditarily in individuals that are there. My wife has 5 biological children. She and her ex. I mean, you can tell they're siblings. They don't necessarily. 2 of the boys look pretty similar, I would say. Yes? Am I missing something? No, good. Sorry. Wasn't that funny? I have one you just saw there. I have 3 children, no biological relationship to myself or my ex at all, because they're all adopted. So they don't have the luxury of knowing a whole lot. But generally, when you adopt children, you don't get a whole picture about those things, unless it's what they call an open adoption, and that has their limitations. That's a whole other series of lectures. Okay? It's hard for my kids to even get information because a lot of the records are sealed. Moving on. So, and this is the whole idea of transmission of traits. Okay? So, this book, this part of the chapter deals a lot with stuff we're not going to do. My focus point is to understand different kinds of mutations. What is going on? What are you doing? We have to understand different kinds of mutations and how they're categorized. And then more importantly, the way that bacteria are able to magically mutate and pass traits on called lateral or horizontal passing of traits when ours is vertical. You and I, where do we get our traits from? Adoptive or otherwise, mom and dad. Okay. That's it. Do they all get expressed the same way? No, because there's so much genetic diversity. However, okay, in that type of thing, lateral, being able to do things laterally or horizontal means, and this is what bacteria can do. If you're all Staph aureus and one of you is MRSA, that person could touch the next person, and then they could touch the next person, et cetera, like a little chain, like when little kids playing the game of telephone. And that whole population overnight is MRSA. That's how rapidly bacteria can change. So it's terribly important. There's 4 major methods that we'll learn about. I'll tell you the names now. You'll see them in print in the time we have left, possibly not. And they are something called transformation, which is sort of a generic term for changing. Not the same as something called transduction, which is your introduction to viruses. Okay, something called conjugation, which is very common. That's the one with the MRSA. That's when they actually touch each other and use that F. pilus or pilus that you might remember we talked about earlier. They thought it was fertility. That was some type of sexual reproduction. It's not. It's just a passageway for genetic material. And a funny one called transposition, which is more significant than you might think. It's almost like genetic sequences jumping in and out around a chromosome. And I have a series of very interesting animations from, and I'm sure they're available in both textbooks since they're McGraw-Hill Properties is the publisher. that will help us with this. Pardon me. So all that type of stuff. So the genome is the total sum, chromosomes mostly. The oddball thing about eukarya, and it's not that relevant here, is plants have mitochondria and chloroplasts. We have mitochondria that have that bacterial genetic material. So in our cells, whether it's our cells or bacterial cells, it's all DNA. The viruses are the oddball. When we get to the viruses, you'll see the instability of RNA reproduction is significant. That's why COVID, influenza keep changing. So there's not a whole lot to say that you don't know already about that. Eukaryotic chromosomes are just that, pairs in the nucleus. So by definition, a normal cell, other than an egg or a sperm cell, is diploid. Haploid is what bacteria is. You have one strand. Doesn't matter if it's a giant circle or a small linear strand, it's still a single strand. That's what an egg and a sperm cell have 23 of those single strands. Inherently, they are more likely to mutate. It is why if you go in for an x-ray, why they're always asking women if they're pregnant, they're always asking, depending on where the location, they're going to have to shield people. Maybe you go to the dentist and get x-rays. Maybe they're putting in a lead apron. We had several lead aprons in my office. We have lead on the walls to prevent scatter because you have to be certified to have an x-ray unit in an office. So you go to a doctor's office or you go to a dentist's office or you're in the x-ray department in the hospital. All of the above. So the interesting thing about bacteria is they're haploid. They're already supercoiled, which makes them more liable to damage. Okay? And they're haploid already. So it's only a single circular chromosome. So already it's a problem, sort of an accident looking to happen. And so we'll look more at that. So genes, we think about genes being all the characteristics. Again, there's a lot of regulatory genes. A disproportionately large number in eukarya. Bacteria, not so many. Most of those are expressed. So if we could analyze all the genetic loci, if we did DNA testing, we could see similar genotypes. But it doesn't necessarily account for things that are measurable or observable, which are phenotypes. And they have some illustrations here, and they're going to talk about plasmids in a bit. We're not going to go into, because this is AP1, or introductory bio for me. Yes, it's got a nitrogenous base, and deoxyribose and phosphate and linkages, and things like, and you didn't do 5' and 3', did you? No idea what the heck I'm talking about. You did? Good. But we don't need to know. That's molecular biology. It is, understand that DNA, they're what they call anti-parallel strands. One's right side up, one's upside down. And when we replicate DNA or make proteins, it's directional. And they go from one direction, like 5' to 3', based on their structure. And so one strand does it continuously, and the other strand makes it in little chunks that you have to splice together. That, if you were taking the micro course for bio majors and pre-meds, that's the kind of stuff you would be. I don't think it's relevant too much for 2370 for this course. And again, the A's, T's, G's, and C's. This is the big deal. I'm sure you learned about it when you looked at the structure of DNA. It's this. It's how they're linked together. Adenine has 2 of these basic units. Okay, so it's got these sort of 6-pack or 5-pack of molecules, carbon bases in a circular chain, somewhat, or in this interesting structure, hexagonal and pentagonal. And this one's a hexagon, and you only have room for 3. And so that's why we have A's and T's, G's and C's. They fit into that structure. That was the famous experiment that I hope you went over in AP1 with Rosalind Franklin. Did you talk about Watson and Crick and how DNA was discovered? they knew historically, and it underscores why women and their role in medicine and science was horribly mistreated for forever and still. No other way to say it. People can yell at me all they want, it's the truth. And she was working with Watson and Crick and another scientist. Those three got Nobel Prizes. She was trying, they knew about adenine, thymine, guanine, and cytosine. They knew the proportions. They couldn't make the molecule to do what they thought it did. She took DNA and said she was trying to X-ray it to measure the space here so she could figure out what linked them. The trouble is she couldn't get the molecule to stop moving. So she put it in liquid nitrogen. So it effectively stopped moving, or moved so minimally she could X-ray it. And she came down one day and said, hey, there's whatever, 15 angstroms, or whatever the number was between them. Okay? And they basically all kind of did this and went on and had their tuna sandwich. Okay? They went and Washington Crick went upstairs and built the molecule out of, for lack of a better word, tinker toys. Okay? And the molecule explained everything. So they published their paper. 5 years later, they get the Nobel Prize. Never mentioned her name. So posthumously, she finally was given her due. Still not a Nobel Prize. They don't award them posthumously. But and it's a interesting scenario to show you the little things are important. That explained the twisted ladder, and these little dots are hydrogen bonds. Don't have to know any of this stuff, because hydrogen bonds open and close, and DNA, for lack of any other term, is a zipper. And biologically, we unravel it when we want to make more DNA, and we re-ravel it when we're done. When we want to make proteins, we unravel it, and we re-ravel it when we're done. Downside up, upside down. Semi-conservative means we keep taking a new strand from the old strand. Not bacteria really don't do that, but they have less problems for whatever reason. So when you open to make, like one cell making two cells, okay, one liver cell wants to make another one, it opens it up, okay, and they use one strand of the old DNA and create one strand of the new DNA. So you always have some fresh DNA with some existing DNA, so it doesn't degrade. Cloning means it's like you photocopied an article, took the photocopied that, took that photocopy, and over time it would degrade. That's what happens. If you've ever seen one of these where you have like a copy that's been copied 63 times, you can barely read it. That's the degradation associated with cloning. And so there's the DNA polymerase 3. So it can proofread effectively. That's what I was trying to demonstrate. With polymerase 1, okay, it'll repair the mismatch. You know, it's as likely to be right or wrong that are there. And this just shows you some of the other stuff. And I don't really care how fast they do it or any of this stuff that's going on there. Not important. When we finally get to the language of DNA, DNA has basically, it has 64 possible 3-letter sequences called triplets. We read them not in DNA, not in A's, T's, C's, and G's. We read them in RNA. And the only difference is uracil replaces thymine. They're almost identical structurally. But for whatever reason, that's how it's done. So when you see, we call the 3-letter sequences, which are A, G, C, and U, codons. We even reverse them for transfer RNA, which helps to assemble proteins. They're called anticodons. To me, the important part is not this, is what's coming up, not that. I didn't mean to do that. Sorry. And it's not so much in transcription, like I said, making the messenger RNA or any RNA, and translation, expressing it as proteins. So none of this stuff, none of that stuff, none of that stuff. God, that looks horrible. That's a transfer RNA, very unusual. So here's a codon, and it's mated to what you would find as an anticodon over here. That's how this picks up, attaches to amino acids, make sure it drags them to the right. We understand it. You don't have to. Just as long as it works for you. right? Works for me. 70 versus 80S did that. Not going to do that. Going to do this. This is the genetic code. Terms of messenger RNA or any RNA. Very simple. There is built-in redundancy. We have 20 amino acids. We have 64 codes. So surely there's anywhere between as little as 2 and as many as 6 codes for the same amino acid. We have exactly 4 punctuation marks. We have a start code and 3 stop codes. We hit a start code, we start making. When we hit a stop code, we stop. However long it is, that's either the polypeptide or a protein. What's the difference? How long they are. Polypeptides are less than 100 amino acids. Proteins are 100 or more. It's not hard. We use the terms almost interchangeably. So redundancy is the great thing about the human body. We have redundancy going everywhere. You have 2 kidneys. You can donate one. And if the other one doesn't work as well, as long as you have 25% of your one kidney working, you're not going to need dialysis. So you have like 7 times or 8 times more kidney than you really need. I mean, it's nice because and that's redundancy. Redundancy is a good news and a bad news type of thing. We have so much of it. The trouble is sometimes we don't know when we're sick, and then all of a sudden we're very sick. That happens with people who breathe fine, all of a sudden are told they have to be on oxygen for the rest of their lives. People who never had any trouble with kidney problems, all of a sudden told they have to be on dialysis, because there's so many built-in safeguards that when they finally go, it's like, again, falling off a cliff. So, and there's a thing here that you're going to see shortly called wobble. Normally, I talk about being forgiven. So if you typically get the first 2 letters right, you don't really have to worry about the third one. Even if it's a mistake, you probably get the right amino acid. And that's the code. I don't have any questions about that. I'm trying to be upfront with you. So you don't see your memorize that? No. So basically, most of these, if you get the first 2 letters, you're good to go. The start code is always this. And if you encounter it after the start, it's a slightly different version of this. You don't know any of the names, amino acids. You have 2 stop codes here and 1 stop code here. That's everything else is fairly straightforward. So here you have 4 possible codes for this. And you get 2 more, 6 for 1. So everything has at least 2. You don't have to know the names. This is just trying to acquaint you with the concept of redundancy. So, I mean, if you make an error in DNA, a mutation and the first 2 letters are C, and it should be a U, but you put a C in, is there going to be any problem? No. We call that an isolated mutation. It's called a point mutation. They're the most common. The fact that it has absolutely no effect makes it a, you should guess it, silent mutation. Tree, falls, forest, sound, right? Silent mutation. We make silent mutations a lot. So if it doesn't affect anything, not a big deal. If for some reason you were over here and you were supposed to be a U and you put another A in, now you would substitute for asparagine lysine. Well, that's different. Is it going to have any effect? I don't know. But we call that a missense mutation. It alters the sense of the protein. Will it affect its functioning? Maybe, maybe not. The most famous single missense mutation is very famous. It's because there's 1 error of amino acid incorporation in sickle cell hemoglobin. 1 error along the way, probably 1 isolated point, creates a potentially deadly disease that up until recently is very, very difficult to treat. And even it still has its limitation. I mean, I worked out of a sickle cell clinic in Richmond for a summer. There's a lot of people with pain. And a lot of people, we were working on their leg sores and wrapping them. Oh my goodness gracious. My mentor there was doing a rotation with, he ran that clinic at the medical school there, Medical College of Virginia. It's Virginia Commonwealth University. And so that's missense. Nonsense is if you prematurely hit a stop code. There's supposed to be 25 in a polypeptide, and you only get 22, and there's a mutation that causes you to hit a stop code. Well, guess what? That's a nonsense mutation. Normally, those are bad because the protein will not work. And if you have enough proteins that don't work, there's going to be problems physiologically. Somebody's likely not to survive. So that's desirable if you're addressing a bacteria with radiation, not so good for us. And these are just some, you will see these, you probably will see these when you do AP2. You'll take a look at how antibodies are made so vigorously is because they're something called polysome. You have one strand of messenger RNA attached to a bunch of ribosomes, so they make it super fast. You don't have to know anything about that. Start codon is always, it's a different kind of methionine, we might use it later, but the one that starts is a particular variation. And that's really what we have out of this particular thing that's here. There's other things that we see, and I'm not getting into it. Classic micro. I used to have to teach this to nursing students. It was totally confusing. It's called the lac operon. Did they do it for your imaging? Anything mentioned about that? It's basically kind of things that govern positive and negative feedback. Okay, it's a classic system we see in E. coli that helps us understand positive and negative feedback. I don't do it because it basically involves these play a significant role in cancers in us, in our cells, promoters and operators. But effectively, it means that in the presence of a certain substance, it'll allow a path to go, and in the absence of that substance, it'll put a blocker. There's the blocker. When something's in the area, then it'll release the blocker and things can work. So I don't really want to go into specifics of the lac operon. I taught a course for years at a community college where we had to go over. Didn't think it was really a good idea. Okay. These become significant when we get into the diseases. Some of those regulatory proteins alter certain traits. And when we get into gonorrhea, it adheres because it makes these fimbriae. So the nasty kind is this, and this is the most common kind. The Streptococcus pneumonia that causes bacterial pneumonia, and really where we learned about transformation, has a capsule. Those big capsules are hard to swallow by macrophages, big player in pneumonia. And we're sort of getting to the point where we'll conclude. Recombination is basically taking on different DNA and incorporating it. So you'll see that in genetic engineering, we like recombinant DNA. We use E. coli or yeasts that are willing to take on and not damage incoming DNA. Okay, and effectively when it's incorporated, now it's changed genetically, so it's a new strain. So we hear genetically modified organisms, that's really the process that's there. And so in bacteria, you heard me mention this before, we take plasmids that we see mostly in bacteria and sometimes in yeasts, and splice codes into them and transfer them into particularly receptive organisms to make different materials, different proteins that we harvest. So any organism that contains genes that originated from another would be recombinant. So even though it's not an organism, the analogy would be COVID. COVID it's not an organism, but it had genetic material from multiple species. So in viruses, we don't use the term recombinant, we call it a novel or new virus, unexpected. It's because it had bat in it, it had pig in it, it had bird in it, it had people in it. And all of that is something so new to our system, we couldn't respond to it other than very irregularly. Now, COVID is starting to wane, and the mutations are more easy to deal with. So those viruses used to be just a cold or a bad cold. So the plasmids, don't have to review that again. Circle pieces of DNA, not useful, but not essential. Necessary for survival is essential. Useful traits, resistance, making a toxin, making a capsule, etc. These are the ones we're going to start to look at next time. Conjugation, transformation, transduction, and another one called transposition. We'll stop there. We'll see everybody Wednesday. Bring back your things.